Namaste students, I am Dr. Aziz Mahmood Khan from Department of Food and Biotechnology, Faculty of Agriculture and Veterinary Science, Jyoti Vidyapith Women's University, Jaipur. Today I am going to discuss about the classification of cytokines and different classes of the cytokines. So cytokines as our nomenclature is conducted, they are also classified on the same basis, their function and the cells from which they are secreted. So the first one is interleukin, second one is lymphokines, third one is monokines, fourth one is interferon, and then fifth colony stimulating factor, and then chemokines. Interleukins are the mediators between the leukocytes, lymphokine secreted by the lymphocytes, monokines secreted by the monokines, interferon have antiviral activity, colony stimulating factor have the growth factor in the blood cells, chemokines are the molecules which mediate chemoattraction and the chemotaxis. First, discuss about the lymphocytes. What are the lymphocytes? Lymphocytes are a subset of a cytokines which are produced by a type of immune cells which are known as the lymphocytes. So lymphocytes secreted by the lymphokines. Lymphokines secreted by the lymphocytes. And they are protein mediators. Each cytokines are some protein mediators. Lymphokines are protein mediators which are produced by the T cells, and they direct the immune system response by signaling between the cells, between the another cells. And the lymphokines have many roles, including attraction of the immune cells, including attraction of macrophages and other lymphocytes to and site and their subsequent activation to prepare them to mount an immune response. So first one is they attract immune cells. Where they attract the immune cells, they attract the immune cells to the site of the infection. And after infection, after attraction, and after to reaching the cells to the site of infection, they perform their activation process also. They perform the uh, differentiation or as well as they perform their proliferation activities also. So these are the functions of the lymphokines, the lymphocyte. Circulating lymphocytes can detect a very small amount of the lymphocyte and then move up the concentration gradient towards where the immune cell response is required. That very small concentration is enough to perform, and they move from the place where their concentration is lower, sorry, higher to the lower, means concentration gradients. On the basis of their concentration gradient, they move from the site of their secretion to the site where there are not any kind of lymphokines. So lymphokines and B cells produce antibodies. So they have also important role in the antibody productions. Now monokines. Discuss about the monokines. The monokine is a type of cytokine which are produced primarily. So these are the molecules which are produced by monocytes and macrophages. Example include the interleukin 1 and the tumor necrosis factor alpha are the two monokines. The other monokines include the alpha and beta and interferon and colony stimulating factor are also the some monokines which are secreted by the monocytes and the macrophages. We know that the monocytes and macrophages have important role in the phagocytosis. They have important role in the phagocytosis. So monocytes and monophages secrete monokines, so they also have important role in the phagocytosis process and the processing of the antigen molecule. They have important role in the processing of the antigenic molecules. Now we talk about the interleukins. Interleukin is a very large class and a very important class also, we can say. And we have taken the examples of the interleukin also. What are the interleukins? Interleukins are the intermediates between the leukocytes. A variety of uh, naturally occurring polypeptides that are family of the cytokines which affect function of special cell types and are found in small quantities are known as the interleukins. They are secreted regulatory proteins produced by the lymphocytes. Okay, and they are produced by the monocytes. They are also produced by some another cells. We know the interleukins secreted by the T helper cells. We have taken the examples also the interleukins from the T helper cells, and many examples were there: interleukin 1, interleukin 2, interleukin 3, interleukin 4, interleukin 5, 6. So these are the examples, and they are released by cells in response to antigenic and non-antigenic stimuli. They consist of interleukin from interleukin 1 to interleukin 37. And interleukin 1 activates antigen presenting cells and CD4 plus lymphocytes. It affects, we are talking the exam functions of each interleukins here. Function and classification and function with together. Interleukin 1 activates the antigen presenting cell and CD4 plus lymphocytes. They affect the differentiation of the B cell and T cell and the immunocomponent cells and takes part in the regulation of the production of other cytokines. Next is interleukin 2. Interleukin 2 we have already discussed in the topic and redundancy. So they stimulate the proliferation of the B cell. They have important role in the B cell. They are we have understand these interleukin 2 in the redundancy also. They have redundancy functions, similar functions, uh, and so they stimulate the proliferation and activation of the B cells and also the T cells. Next is interleukin 4. Interleukin 4 plays a role in the differentiation of T helper 2 cell, which in allergic reactions and also in the class switching of the antibody types. There are more functions about the interleukin 4 which we will discuss on. Interleukin 3 is a potent activator of the hemopoietic cells, means nerve cells. Okay, so they are potent activator of the new hemopoietic cells. Stimulates the natural killer cells and act as a synergist with the interleukin 4 during the induction of the CD4 plus lymphocytes activation process. We know about the interleukin 4, no need to talk, they have important role in the activation, position, and differentiation of B cell, uh, thymocytes, and other cells. Interleukin 5 stimulates the production of maturation of eosinophils. Have important role in the production and maturation of the eosinophils during the inflammation process, and also have important role in the uh, important role which functions with the interleukin 4. Interleukin 7 is also known as the growth factor of immature B cell and the T cell. is the growth factor. And it induces the apoptosis of the tumor cells and causes the differentiation of the cells from a subgroup of acute myeloblastic leukemia. Interleukin 8. Interleukin 8 act as a chemotactic effector and attract neutrophils, basophils, and T cells. So if there is any type of inflammation, then interleukin 8 will attract neutrophils, basophils, and T cells to the site of the inflammation. Mm, Interleukin 9 is another interleukin which stimulates the secretion of the interleukin 2, interleukin 4, interleukin 6, interleukin 11, and also takes part in the stimulation of the cytotoxicity of cytotoxic cells and natural killer cells, which go for the apoptosis process. So it has many functions and activation of another uh, interleukins and also in the killing action of the cytotoxic and natural killer cells. Interleukin 10 acts as a repressed secretion of the pro-inflammatory cytokines. So I discuss here some interleukins, interleukin 1 to interleukin 10. Now the next is interferons. What is the function of interferons and what are the interferons? Interferons play an important role in the first line of defense against viral infections. If we talk about uh, the protection of baby, then how the babies are safe. They do not have any kind of third line of defense system. They do not have any kind of well-developed immune system. So they have interferons. And interferons, as we know that they have antiviral activity. And it is very effective. Uh, that is the main reason that the babies are safe from the COVID-19 also. These all is due to these interferons. Uh, Interferons are part of the non-specific immune system and are induced at an early stage in the viral infection before the specific immune system has time to respond. It has early response. And interferons are made by cells in response to an appropriate stimulus and are released into the surrounding medium. They bind to the receptor on target cells and induce transcription of approximately 20 to 30 genes in the target cells, and this results in the antiviral stage. On there are various types of interferons are there, and based on the type of the receptor through which they signal, human interferons can be classified in some three major types: interferon 1, interferon type 2, interferon type 3. All interferon, all type 1 interferon bind to a specific cell surface receptor complex, which is known as the interferon alpha receptor. Interferon type 1 will bind to a particular receptor, which is known as the alpha receptor. Interferon NAR that consists of an IFNAR1 and IFNAR2 genes. Two genes will be there in the interferon type 1. The type 1 interferon present in the humans are interferon alpha, interferon beta, and interferon omega. Three interferon alpha, beta, and omega are included into the type 1 interferon. And interferon type 2, what is added in the interferon type 2, they bind to the IFNGR that consists of IFNGR receptor and IFNGR2 chance receptors. In humans, this is interferon gamma. So interferon gamma is class 2 type. Interferon type 3 signal, so a receptor complex consists of the interleukin 10 R2 and interleukin R1. So they are in that classes. Now we talk about the chemokines. Chemokines are a family of the small cytokines or signal proteins which are secreted by the cells. Means they are small cytokines, and their name is derived from the ability to induce direct chemotaxis. They have the capability to kill the cell directly in the nearby cells, and they are chemotactic cytokines. Proteins are classified as the chemokines according to structure characteristics such as their small size of approximately 8 to 10 kilodaltons, and the, they are also known for their presence of 4 cysteine residues. So these are the most important characteristics of the chemokines, and some classification is also there, which are the subfamilies of the chemokines. CXC chemokine, CC chemokines, CX3C chemokines, and XC chemokines, which have different kinds of receptor families. Now, colony stimulating factor. Colony stimulating factor are secreted by the glycoprotein that bind to receptor proteins on the surface of the hematopoietic cell. Hematopoietic stem cells are the new cells from which another cells are generated, and they activate the intracellular signaling pathway. They can cause the another cells to proliferate and differentiate into specific kind of blood cells. So, if we these colony stimulating factor have important role in the conversion of hematopoietic stem cells into a particular type of cell, particular type of blood cells. It may convert into the blood cell. It may convert into white cell, white cells like granulocyte cells and agranulocyte cells. Any kind of cells can be converted due to the colony stimulating factors. Okay, it depends on the type of the CSF that which cell will be developed, which cell will be differentiated. So the colony stimulating factors are soluble in another, and they are membrane-bound substances on the hematopoietic microenvironment. They translocate by paracrine signaling, endocrine signaling, and also autocrine signaling. And the colony stimulating factor have some examples like CSF1. Uh, they, the CSF1 have macrophage colony stimulating factor. CSF2 is a granulocytes macrophage colony stimulating factor, and CSF3 granulocyte colony stimulating factor. CSF. So these are the colony stimulating factors, and now the tumor necrosis factors. Tumor necrosis factor refer to a group of cytokines that can cause the death. Means they have the capability to kill the cells. They have the capability to kill the tumor cells also. 19 cytokines have been identified as the part of the tumor necrosis factor on the basis of the sequence, function, and structure family. These tumor necrosis factor are formally known as the TNF alpha or TNF alpha is the best known example and the best known member of the tumor necrosis factor. It is very important. If a question arises that which is the most important tumor necrosis factor, so we can answer that TNF alpha. TNF alpha is a monocyte which is derived from the cytotoxin that has the capability to tumor regression, septic shock, and the cachexia. They have capability in the tumor degradation. Lymphotoxin alpha, which is another type of tumor necrosis factor, lymphotoxin alpha, and it is was previously known as the tumor necrosis factor beta, is another cytokines which that is inhibited by the interleukin 10. So it is also important. Lymphotoxin alpha and lymphotoxin beta, two related cytokines, are produced by the lymphocytes that are cytotoxic for a wide range of tumor cells. So if we need to kill any kind of tumor cells, then there can be an answer to killing the tumor cells, that tumor necrosis factor alpha and beta can be used. So my lecture for this is now over. In the next video, I will discuss about the cytokine receptor families. So students will be able to understand the type classification of the uh, cytokines, and if questions arise, which type of questions can be asked by the examiner. The examiner can ask that explain cytokines receptor family, explain uh, types of cytokines, explain the function of the cytokines. So these questions can be asked. So my lecture is over here. Thank you very much.